Hello, and welcome to our presentation on nucleic acid delivery for disease therapy. My name's Belinda, and I will begin by introducing nucleic acid delivery for disease therapy and providing an overview of its purpose, use, history, development, and finishing with how it works before handing over to Zane and Kimberly to talk about current applications of the therapy, advantages, shortcomings, and potential future applications. Nucleic acid delivery for disease therapy is a therapeutic technique which is intended to treat diseases caused by or associated with an abnormal gene expression. This technique uses either nucleic acids themselves or compounds that are closely related to nucleic acids to potentially treat a range of diseases, from diabetes to cancer to infectious diseases. Fundamentally, the use of nucleic acid therapy is intended to stop the manufacturing of diseases related to abnormal proteins, while leaving all other proteins untouched, theoretically treating the genetic cause of a disease. Many traditional therapeutic interventions are not targeted to the disease they are projected to treat, and therefore can result in numerous serious side effects, which can eventually lead to necessary cessation of treatment. By increasing the accuracy in which treatments target diseases, nucleic acid delivery also aims to minimize the side effects experienced with traditional therapeutics, and in turn, allow treatment to continue as required. However, in spite of this potential, integration into clinical settings has been held back by limited bioavailability of nucleic acids and their synthetic analogues in target cells. As a result, many different nucleic acid delivery systems have now been developed in an attempt to overcome this limitation. Nucleic acids have been considered potential therapeutic agents for over 40 decades now. However, early on, the knowledge about this therapy was restricted and narrow. Over the last 3 decades, following diverse research, the field has seen substantial progress in realizing the therapeutic application of nucleic acids. The growing interest in this field can be seen with the numerous scientists and engineers now involved in its study. A number of advances have been pivotal in this progress. The identification of several molecular pathways central in the study of diseases. The discovery of additional types of RNA, including microRNA and short interfering RNA, each with complex biological functions essential to nucleic acid therapeutics. The discovery that RNAs can behave like enzymes, which has led to progress being made regarding the development of RNAs with additional properties that are useful for therapeutic applications. And finally, the enhancement of the bioavailability of nucleic acid-based therapeutics. All of these have been made possible by the technological advances in DNA synthesis, including the use of DNA shuffling, combinatorial chemistry, genetic engineering of viruses, synthesis of longer DNA constructs, bioprospecting, and high-throughput screening. Over the last 2 decades, studies regarding nucleic acids have demonstrated exciting potential for their use as therapeutic agents, and as a result have been gaining substantial clinical and scientific interest. Some studies have shown positive results. However, many clinical trials have experienced not so satisfactory results, including difficulties in targeting the cell's cytosol or nucleus for treatment and some unexpected severe adverse reactions. There have also been some safety and ethical concerns due to the intended treatment of modification of the expression of genes. Of the numerous nucleic acid therapies, though, several are now in clinical development, and regulatory agencies have also approved several more for clinical use in humans, especially when used to treat severe diseases such as AIDS and cancer, where the benefit of the treatment outweighs the potential risk to the patient. However, ongoing current research is focused on improving the efficiency of nucleic acid transfection prior to allowing the remaining therapies to proceed to human clinical trials. Nucleic acids are hydrophilic, negatively charged, and quite large molecules, which means that by themselves they do not easily enter the cytosol or nucleus of a cell. In order to reach the cell, they also have to cross physiological barriers and are therefore vulnerable to both intra- and extracellular enzymatic degradations. These issues have led to the essential development of delivery systems in order to assist the nucleic acid in reaching its target cell. There are many applications of nucleic acid delivery which will be discussed later in this presentation. However, a basic overview is that nucleic acid is usually delivered to a cell with the aid of a vector, which is generally a virus. These vectors need to have the ability to attach and enter the target cell and transfer the nucleic acid to the cell. Once in the cell, the cytosol or nucleus, the nucleic acid treats diseases by interfering with either the transcription or translation of an abnormal or non-functional gene's mRNA or protein, and in doing so amends its expression. As a result, the nucleic acid re-establishes the correct functioning of the cell and potentially treats the genetic cause of the disease. I'll now hand over to Zane, who will be speaking about the current applications and advantages of nucleic acid delivery for disease therapy.
Current applications of nucleic acid delivery. Nucleic acids such as plasmid DNA, antisense oligonucleotides, and RNA interference molecules have a great potential to be used as therapeutic for a treatment of various genetic and acquired diseases. To design a successful nucleic acid delivery system, the pharmacochemical effects of nucleic acid, the physiological condition of the subject or sites, and the physicochemical properties of nucleic acid and carriers had to be closely examined. Viral and non-viral vectors have been most effective for delivery of nucleic acids and are used in current clinical trials due to the recent developments in their design and safety. Non-viral carriers or synthetic vector delivery systems offer several advantages that have significantly advanced in their development. These include improved biosafety and flexibility. They are also simpler to manufacture and modify compared to the viral vectors. The most basic approach is to increase the expression of a gene of interest by incorporating into viral vectors or bacterial plasmids and expressing it to the tissue or cell to restore normal or correct for pathological gene expression. Polymer carriers are promising vectors for gene delivery due to their potential for structural diversity and flexible functionality. They could enhance the complexes of nucleic acids, stability during the circulation, and give a great imaging potential. Cell-penetrating peptides, CPPs, have been demonstrated to allow for efficient delivery of nucleic acids and their oligonucleotide analogues. In cell culture in short delivery peptides, CPPs are cationic and amplified peptides, usually 30 amino acids in length. They have been categorized by their remarkable ability to convey different cargo molecules into cells and tissues, Importantly, they have demonstrated excellent potential for the delivery of nucleic acids, ranging from natural or synthetic short RNA and DNA oligonucleotide, much larger plasmid molecules, both in vitro and vivo techniques. Several additional gene therapeutic platforms have emerged, such as gene editing platform, zinc finger nucleases, and transcription activator-like factors, nucleases, and so on, for accessing and alerting the genomic DNA, but also chemically stabilized mRNAs that could be used for templates for protein synthesis and unmodified mRNA. Advantages of nucleic acid delivery. Delivery methods must consider half-life in the bloodstream, tissue specificity, toxicity, and efficiency. Some proposed methods include topical delivery of gold nanoparticle nucleic acid conjugates throughout the skin, and encapsulating nucleic acid molecules with liposomes. These can travel safely throughout the bloodstream, to be taken into a tumor or infected cell by fussing with a cell membrane, and then depositing the molecule inside the cell. There are many advantages of nucleic acid delivery, just to name a few. Increase in treatment possibility for genetic and inflammatory diseases. The innate immune system distinguishes damage-associated molecule patterns or pattern-associated molecule patterns as the release of nucleic acid waste from dead or dying cells. Arousal of pathological inflammation and autoimmune disease may occur if the innate immune system is improperly activated. Pattern recognition receptors such as toll-like receptors that can distinguish the damage-associated molecule patterns or pattern-associated molecule patterns, or the downstream effector molecules they arouse, have had much focus on beneficially targeting and limiting inflammatory and preventing these diseases. The capacity of the activation of pattern recognition receptors by the nucleic acid ligases can be limited by nucleic acid binding polymerase acting as a molecule scavengers. Another important nucleic acid therapy advantages include precise or transcriptional targeting of tissue and the ability to completely toxic properties in proteins with encoded genes. Resistant mechanisms, such as those established by P-glycoprotein, can be overcome by nucleic acid macromolecules. In blood circulation, nucleic acid must be condensed into polyplexes to guard against their vulnerability to enzyme degradation. Threats aimed at nanosystem by physiological barriers such as reticuloendothelial system must be guarded against contact with undesirable blood cells, resulting in unwanted blood vessel adhesion or antibodies and complementary proteins that cause pathogens to become more susceptible to phagocytosis. Shielding effects by polyethylene glycol of nanosystem can assist in cell targeting and internalization alongside decorated ligands binding to receptors. Now I'll pass on to Kimberly to talk about shortcomings and potential future applications of nucleic acid delivery.
Since the support of the first antisense drug was approved, there has been much progress in the field of oligonucleotide therapies. With the use of siRNA and splice switching in late-stage clinical trials, there have been hopeful developments. With this being said, a major issue is the effective delivery of oligonucleotides to their intracellular sites. Oligonucleotide-based therapies has the key problem of delivering the active oligonucleotide to its site of action in the cytosol or the nucleus of the cells within tissues. The first issue is delivering the oligonucleotide to the tissue while minimizing exposure to the other tissues. The other issue is being able to deliver the oligonucleotide to the correct intracellular compartment. The following image shows the tissue barriers to oligonucleotide delivery. The star shapes represent free oligonucleotides and the blue circles represent oligonucleotides in nanoparticles. If we look at image A in the top right-hand corner, we can see the blood-brain barrier has tight endothelial cells, as well as pericyte and astrocyte processes which make it impermeable for free oligonucleotides and oligonucleotides in nanoparticles. In image B, on the bottom right-hand corner, we see the blood tissue barrier, which allows for many oligonucleotides to cross the endothelium via diffusion. Image C, in the bottom left-hand corner, is the blood-liver barrier. We can see the fenestrated endothelium in the liver and spleen is easily permeable by both free oligonucleotides and nanoparticles. The importance of the different barriers will depend on the physical as well as chemical properties of the oligonucleotide being used. For example, when a type of nanocarrier is used, the biodistribution of antisense and siRNA oligonucleotides used as individual molecules would be very different. Another shortcoming is the fast clearance by the kidney. The kidneys play an important role in the pharmacokinetics and biodistribution of oligonucleotides. Usually, molecules which are 3 to 6 nanometers or less in size can be ultrafiltered by the kidney. As many types of oligonucleotides fall in this size range, they can be quickly excreted by the renal route. Due to their low affinity in binding the plasma proteins, renal excretion can occur rapidly for siRNA and uncharged oligonucleotides, and they tend to accumulate at lower levels in tissues. Renal excretion of antisense oligonucleotides is reduced in plasma protein binding and allows for future distribution throughout the body, compared to siRNA. Phosphonothioate and antisense oligonucleotides bind to plasma proteins, consequently slowing renal clearance and enabling wide-ranging distribution to tissues with high accumulation in the liver and kidneys. Other problems can include adverse reactions with blood components or with intracellular proteins. Many strategies are currently being studied for future applications of nucleic acid delivery for disease therapy. One is to incorporate the oligonucleotide into a nanocarrier, and another one is to chemically modify the oligonucleotide itself. This would be done by targeting the ligand while maintaining the molecular nature of the conjugate. While the conjugate approach is still in its early days, there is a promising path for oligonucleotide therapies. There have been much interest in the molecular scale ligand oligonucleotide conjugate, which allows the prospect of selective delivery to specific cells or tissues via receptor-mediated mechanisms, together with the prevention of the toxicities often associated with nanocarriers. Another development is the blood-brain barrier and therapy of central nervous system diseases. Tricyclic modification provides increased affinity, but it is still unclear why molecules should cross the blood-brain barrier while other oligonucleotides with similar backbones do not. Systemic delivery of oligonucleotides to the central nervous system remains a challenge and are still unresolved. In the context of cell therapies, in vitro transcribed mRNAs combining modified bases can effectively express proteins in vivo while having reduced effects on the innate immune system.